Hello, everybody. Welcome back. We are going to be covering Chapter 10 in this lecture, which is focusing on meiosis, a type of cell division that is important in sexual reproduction. So we introduced different types of reproduction a little bit in Chapter 9 with asexual organisms, just making a copy of their DNA and splitting it into like a clone, whereas sexual reproduction, where you actually combine genetic information from two different organisms to create a genetically unique offspring. So we're going to take a look at how meiosis plays a role in this process of sexual reproduction. All right, so while there's a lot of organisms that do asexual reproduction, there's quite a few more organisms that have the ability to do sexual reproduction. Almost all plants, animals, fungi, a lot of protists do sexual reproduction. Even some prokaryotes, who are typically just doing that binary fission, have the ability to exchange genes. It's not a reproductive process, but this advantage for organisms to exchange genes, primarily in the sexual reproduction, it's so widespread, so there has to be some kind of advantage to that. And there is one, and it is genetic variability. So this is the big payoff, if you will, for all of the complex things that have to take place for you to get genetically unique offspring. Gives us advantages in evolutionary ideas of survival of the fittest, natural selection. The traits that are best suited for the environment get passed on, and you get those traits by these genetic recombinations that come through meiosis. All right, so why is this important? So I kind of answered that question. This DNA reshuffling is advantageous evolutionarily because it offers the ability for nature to select the traits that are best suited for that environment. So you have some examples here, some plant examples. So what I've shown you here are potatoes. These are just your regular russet potatoes you might find in an Idaho field. And if you'll notice, they're all pretty much the same. They are the same species. You will find fields and fields and fields of these particular types of potatoes on farms in the United States. And one of the major customers for these types of potatoes, you might guess, McDonald's, right? McDonald's, this franchise, this chain all over the world, would like to have all of their French fries look the same color, have about the same length. And so to do that, they have to have all of their French fries be cut from the same potato, which is great for McDonald's French fries and great for the farmers. However, there's some drawback to having all the plants on your farm be identical. This is what we call monoculture, where you have one species of plant being farmed in a particular location. The downside to that is if every plant is pretty much the same, there might be a slight genetic variation, but if you're all growing the same potato, what if a pest comes along, or a pathogen like a virus or a fungus comes along? All of those potatoes are susceptible to that particular infection or virus or disease that comes through. So when you have genetic variation, there might be some individuals in your population that are stronger than others, and that's going to allow them to survive that threat or that challenge to health. Now, if you take a look at the potatoes that are farmed down in South America, I think these are Peruvian potatoes, you'll see there's quite a bit of diversity. They don't really look like our Americanized potatoes, if you will. And so they have a lot better outcome, and they don't have to work as hard of doing pesticides and herbicides and all of these different things that some westernized farmers have to deal with, because you have a variety of plants. This is called polyculture. So monoculture versus polyculture farms is that you are trying to recreate more of the natural environment. You have different organisms, different types of potatoes, tomatoes, corn, all these different things that some might be stronger than others in certain conditions, where others could be stronger in slightly different conditions. So no matter what's happening in the environment, you're not going to completely lose your entire crop. So genetic variability is very important in potatoes as well as all living things. So you can just take that idea of monoculture and polyculture and kind of translate that onto humans. When you have everybody, if everybody was exactly the same with no variability, one pathogen could come in and wipe out the whole population. So genetic variability and diversity is a good thing for populations. Okay, so genetic information, this information we're trying to pass on, shuffle around and pass on to our offspring, is found in chromosomes. So we saw that a little bit back in chapter 9 with our mitosis lecture we just got done with, is your chromosomes are big pieces of DNA. If you remember from our chapter 3, organic molecules, DNA is the instructions, the blueprint for how a cell is a cell, and it is encoded for in the sequences of those nucleotides. So if you remember A's, T's, C's, and G's. Now, when we are taking a look at sexually reproducing organisms, they will typically have, there's a few exceptions, but we'll just kind of go with the typical or arrangement. They will have 2 copies of each chromosome. So here I have a picture of a green and a purple chromosome. They would be carrying all of the same genes. So maybe the genes for hair color or eye color or liver enzymes or hormone production, whatever the instructions are for that particular cell on that chromosome, all of the genes in the green chromosome are the same as the genes on the purple chromosome. They might have different expressions, which we'll see here in a little bit, but they're basically all of the same instructions. Now, in sexually reproducing organisms, they have 2 of each of one kind of chromosome, and we have a special term for that. It's called homologous chromosomes. So homologous chromosomes are kind of partners, the same chromosome, they're carrying the same genes, but typically one came from one parent and one came from the other parent, right? So you have the representation of the genetic information from both of your parents in every one of your body cells. We'll be defining that word here in a little bit. So when we are talking about these homologous pairs or homologous chromosomes, it just means that we are looking at the same chromosome, but one came from mom and one came from dad. They have the same genes, but came from different sources. But that now is your genetic makeup because you are the offspring of the coming together of the egg and sperm. All right, so what we have here is actually what's called a karyotype, this big rainbow picture of chromosomes. This is a picture representation of a human chromosome, or sorry, human complement. This is the genome of a human. So humans have 46 total chromosomes, 23 pairs, if you will. So there's 23 sets of chromosomes. So a lot of times you'll see the chromosomes in this replicated form. So remember when we were talking about mitosis in the S phase of interphase, DNA goes from a single unreplicated chromosome. So over here, so this is the picture I just had. So here's a single unreplicated chromosome, that's the green one, and a single unreplicated purple one. These are homologous pairs, they're partners, they have the same genes on them. Then they go through the S phase of interphase, and now this is one chromosome, kind of they have the bracket here. So this is one chromosome of the green, and this is one chromosome of the green. We just now call it a replicated chromosome, because if you remember, we have our centromere in the middle, and these are our sister chromatids. They're just the replicated pieces of the DNA. It's not 2 chromosomes. We don't have 2 green chromosomes there. We have one green chromosome and one purple chromosome. We have 2 replicated chromosomes, and because they carry the same genetic information, we call them homologous chromosomes. Okay, so hopefully you're kind of getting a feel for that vocabulary. So here in this, the picture kind of the karyotype, these rainbows, rainbow chromosomes, this is a map of basically all of our 23 pairs of chromosomes. So they are named numerically 1 through 22, and then our 23rd set is our XY. These are our sex chromosomes. So for example, chromosome number 1, we have 2 chromosome number 1s. So you've got 1 chromosome number 1 from your mom, 1 chromosome number 1 from your dad. That's what this picture is representing. Here's the centromere in the middle. Here's your sister chromatids here and here. This is one replicated chromosome and its homologous partner in the replicated form. All right, so that's chromosome pair number 1. And then we have chromosome pair number 2, number 3, number 4, so forth and so on. And each one of these chromosomes carry different sets of genes. The homologues, right? So if we say on, say, chromosome number 2, This right here, I'm going to draw this red line. Let's say that's the gene for taco tongue. You guys know what taco tongue is? Like this. All right, can you do that? There you go. If you can roll your tongue into a U shape, you have the gene that allows you to do that. And let's say that's found in this upper arm of chromosome number 2. Well, let's say dad is going to be in this red color. So that's where I'm putting the red stripes on that chromosome number 2. That's going to be the dad's version of that gene to give you the ability to do taco tongue. Now on the mom chromosome, she's going to have the same gene, taco tongue gene, but maybe mom can't do taco tongue. So that's her version of the gene. So both chromosomes, the red one from dad and the green one from mom, are both carrying the gene for taco tongue at the exact same position because it's the same chromosome, but they might have a mom version and a dad version, right? So we're going to get to that. We're going to call that something here in a little bit. So as you are recognizing this homologous pairs, that's super important in understanding meiosis, is that these are replicated copies when you see them in that X pattern of homologous pairs. And each one of these pairs is going to have their own unique set of genes that code for different things that the cell has to do. And every single cell in your body, except your egg and sperm, have this going on in the nucleus. You have 23 pairs of those chromosomes. Now, they're not always going to be replicated like this. They're only going to be replicated in S phase, G1, prophase, metaphase, and then in anaphase, they get pulled apart, right? So we just got done with that in mitosis chapter. So this is not how they're always going to be in your cells. They will just be individual unreplicated chromosomes in G1 and, anaphase and telophase, and cytokinesis in G1. So go back to the cell cycle to help kind of figure that out. So I wanted to talk a little bit about these down here in the corner. So these are called our sex chromosomes. So usually homologous chromosomes, kind of their definition is they're going to have the same genes. I just told you that with the example of chromosome number 2. But your sex chromosomes aren't always homologous. This is a karyotype of a typical male. So when you are talking about your X and Y chromosomes, XY combination, this is tends to be a male. And then we have XX chromosomes, that will be a biological female. Now, when you have XX, those would truly be homologous pairs because they're both X chromosomes, they're carrying the same genes, maybe different versions of the genes, but the same genes are located in the same place. Now, clearly, an X chromosome and a Y chromosome are not truly homologous, but they have to partner up because they can't partner up with anybody else. So your X and Ys do partner up. They are considered a pair. They're just not technically a homologous pair because X chromosomes carry different genes than Y chromosomes. Okay, now do all of our cells, I just mentioned all of our cells with the exception of our egg and sperm, do they all have homologous chromosomes? And the answer is no, not all of them. So let's define a couple terms here. Or locations of these different types of cells. So we have a word called somatic body cells. So somatic body cells are basically everything in your body except your egg and sperm. So if you are a female, every cell in your body except the eggs in your ovaries are considered somatic body cells. For males, everything in your body is a somatic body cell except for your sperm cells. These somatic body cells, right, everybody but the gametes will have homologous pairs. You're going to have two of every chromosome in those somatic cells. Now, your reproductive cells, these are also called gametes. These are your eggs and your sperm. They will only have one pair, right? So they're not doubled up. So all your somatic body cells are doubled up. Your sex cells, your gametes, are not doubled up. They just have one of each. So one number 2, all the way to 1X or 1Y chromosome. Now we have some terms that kind of help us, right? Always new vocabulary. So when you have two sets of your chromosome number, so we have 46 total chromosomes, we have 2 of 23 different kinds, right? So 2 sets of 23 gives us 46. So when you have 2 copies of each chromosome, we use the word diploid. Okay? Diploid, di meaning 2, ploid means chromosome number. So diploid is you have homologous chromosomes. And a lot of times you'll see that indicated with this little abbreviation of the 2N. 2 is representing for the di. the N is representing your set of chromosomes. So you have 2 sets of chromosomes for your 2N diploid. So in this diploid representation here, we've got 2 short, we've got 2 medium, and 2 long, representing the donations from the parents of this particular organism. One from maternal and one from paternal. Now, when we are talking about those sex cells that only have 1 copy of each chromosome, we use the term haploid. So haploid is like half the number of your diploid. So half the number of chromosomes from your somatic cells, there would be no homologous pairs. So in this example over here, we can see there's only 1 short, 1 medium, and 1 long. There's no partner, there's no homologue here. And we would just use the abbreviation N, or sometimes you might see it just 1. And you can use capital or lowercase, I've seen it both. So 1N or N just represents 1 set of the chromosome. And so these are 3 non-homologous chromosomes. Okay, so for animals and humans, as we are kind of under the animal kingdom, we will have almost all the cells in our body are diploid, and only our sex cells, our gametes, egg and sperm, are called haploid. Now there are some organisms out there, not very common in animals, but very common in plants, is they're called polyploid. Poly again meaning many. So some plants have tetraploid or hexaploid, meaning they have 4 sets or 6 sets or some other combination of sets of chromosomes. Now for plants, that's okay. For animals, that is not normal, and usually some abnormalities or diseases can arise from that. We'll talk about that at the end of the lecture today. Okay, so here's those X and Y chromosomes, just a little word about those in more detail. So these are what we call our sex chromosomes. It is the combination of these sex chromosomes when you take your haploid egg carrying an X or a Y and your haploid sperm carrying an X or a Y, and they fuse together to create your diploid zygote. Now, if you get an X and an X, this is the combination that gives you a biological female. If you get an X and a Y, this is the combination that gives you a biological male. So that's the combination of that 23rd set of chromosomes, your XY chromosomes. All the others, 1 through 22, are what we call autosomes. They don't have femaleness or maleness traits or genes associated with them. Those are primarily found on the X and the Y chromosomes. Okay. All right, so again, here's that karyotype showing the X and the Y chromosome. So this is a karyotype of a male because there's an X and a Y chromosome. When you are determining if you have kids or planning on having kids or know somebody who's had kids and they're kind of joking around, are you gonna have a boy or a girl, or dad really wants a boy or dad really wants a girl, or so forth and so on. It really is the sperm that determines the sex of the offspring. So females are XX. So they only donate half of their genetic information. So their eggs only have 1 of their sex chromosome pairs, but they only have X's. So females can only give an X. Males, on the other hand, because they're XY, their sperm only carrying 1 of each of the chromosome, they will carry either the X or the Y. So it is up to the male sperm, which fuses with the female's egg, to determine the sex of the child, whether it's going to be an XX female or an XY male. Okay, so all of those genes that are located on the chromosomes, each chromosome contains hundreds of genes. They're just stacked all on these long sequences of DNA, but each gene is found in a specific location. So like I was drawing the green and the red lines earlier for taco tongue. Now, I don't know if that's exactly the location of the gene on chromosome number 2 for taco tongue, but that was just an example. But there actually is a mapped out physical place on every chromosome for the genes that it contains. And we have, in evolutionary past, there have been some mutations that show up within these genes that can give you a different expression of that gene. So for example, I'll stick with taco tongue. We'll say gene 1 here, down here, the green code, right? The green gene. So let's say that codes for taco tongue. And let's say the light version you can. So I'll do a happy face, right? So you can do taco tongue if you express the light green version of this gene. But if you have the dark green version, then no taco tongue. A little bit of a sad face. You can't do that cool tongue trick. So it's still the same gene in that it's coding for the musculature of your tongue, right? So that's the gene. But we have two different options, if you want to call it that. We have a word for it. They're called alleles. So we have the taco tongue allele and the no taco tongue allele. So this is some vocab that's going to help us when we get to the inheritance chapter coming up a little bit. So you have your different kinds of alleles. So let's say gene 2 codes for something different. Let's say, okay, widow's peak. So I don't have a widow's peak, but some people, their hairline comes down kind of to like a point on their forehead. So that's called a widow's peak, all right? So that is coded for by one gene. So let's say this particular gene, the light blue version, that's horrible. That's my widow's peak. It looks like a little bird. So let's say the light blue gene codes for widow's peak. Now, if both of these homologous chromosomes have that peaky hairline, then that offspring is going to have that trait. They're going to be able to have the widow's peak because that's the only instructions that they have. Whereas for taco tongue, there is two options. There's two alleles. This individual has one that can roll the taco tongue and one that cannot. Now, we'll get into dominant and recessive and what that means when we get to chapter 11 with inheritance. We're not going to talk about that quite yet, but know that it's coming. But just for the purposes of meiosis, it's important to know that genes are located at specific places on the chromosome, and you can have variations within the expression of that gene, and this is what we call alleles. Okay, I have the example here of sickle cell. Hemoglobin. So hemoglobin is a protein. So you could have a gene that codes for your hemoglobin normal and it carries oxygen just fine. But some people have a mutated form of that gene. It's a different allele. We have the normal sickle cell, or sorry, the normal hemoglobin and the mutant sickle cell. Now I actually have the picture of that right here. It's a protein, right? So we see the alpha helices and kind of this big blob of amino acids folded in a particular way. So this is the normal hemoglobin on the top. And if you have that mutation, it's the same gene, codes for your ability to make hemoglobin, but when you have that sickle cell allele, your hemoglobin is folded slightly differently, so it's going to work a little bit less efficiently, a lot less efficiently. We'll see the sickle cell example show back up when we get to the inheritance chapter. It's a good example for dominant and recessive alleles. All right, so all of this stuff, you're probably like, I thought we were going to talk about meiosis. Why do we have to cover all this stuff, homologous chromosomes and alleles and genes and stuff? It is because it is during the process of meiosis and sexual reproduction that we get that genetic diversity, right? That was the whole point of sexual reproduction, is to have offspring that have different traits than their parents and different traits than their siblings and other relations. So during meiosis, those alleles located on the chromosomes are going to get shuffled around and give you some of that genetic diversity. So when you have the combination of an egg and sperm, that zygote, that fertilized egg, is going to be genetically different than the parents. So meiosis is a process that's going to tell us how that shuffling takes place, how that unique genetic combinations end up in the egg and sperm to give us the unique offspring. Okay, so the whole point of meiosis is to create gametes. So gametes are your sex cells, your egg and sperm. So if you're a female, your ovaries are going through the process of what we call oogenesis, which is basically egg making. And if you are a male, your testes go through the process of spermatogenesis, which is sperm making. And so they do that by going through this special kind of cell division. So mitosis, if you remember, was you have one parent cell, you get two daughter cells, they are genetically identical. In meiosis, you take one parent cell, you get 4 daughter cells that are genetically unique. Okay, so that's kind of one of the major differences between these two types of cell division. So this is just a summary, and we're going to go through all of the details here in a little bit. So we have, we'll be talking about meiosis is in 2 rounds, where mitosis was just prophase, metaphase, anaphase, telophase, and we're done. Meiosis is prophase, metaphase, anaphase, telophase 1. And then we have to do it again, prophase, metaphase, anaphase, telophase 2. So we get to go through all of those phases twice for different reasons. It's just not exactly the same. It's not just a copy paste. It's different things that the cell has to do to get to our goal, which are these gametes, these sex cells that have half the number of genetic information and unique combinations of those alleles. So we have just a cell, let's say a cell only has one chromosome set, right? It's got one chromosome, the long chromosome, but it's diploid. It has two of each of the chromosome. So that's the yellow and the purple. That could become from, one came from mom, one came from dad. They have replicated, so they're in that replicated form right there in that X shape that lets us know they've gone through S phase of interphase, and they're lined up. They're homologues because they're going to have the same genes. In the first round of meiosis, what we're going to see is the homologues, the partners, get split. So they don't get to stay next to each other. Then we have our replicated chromosomes. Then the chromatids have to split to get us back to our individual chromosomes. So if we take a look at our 4 daughter cells, if we started off with 2 chromosomes, each daughter cell has 1 chromosome. So we've reduced the number of chromosomes by half. All right, so we're going to go through all the steps that's going to allow us to get to that end point. So we have meiosis is broken up into meiosis 1 and meiosis 2. They will have started with interphase. So these stem cells, if you will, the egg stem cells and the sperm stem cells, they're diploid. They have 2 sets of every chromosome. They've gone through into interphase S1. So we're going to start with replicated chromosomes at this very beginning, prophase 1 of metaphase 1. So just keep in mind, the chromosomes aren't always going to be in that replicated form. They're replicated specifically to begin the process of meiosis. Okay, so let's jump in. So prophase 1, so if you'll take a look, it looks very similar to prophase of mitosis. So when you are practicing your phases, mitosis and meiosis, please make sure you don't forget the 1s and the 2s. It's very important that you are identifying the differences because you have 2 prophases. You actually have 3 prophases. You have mitosis prophase, you have prophase 1 of meiosis, and prophase 2 of meiosis. So don't forget the numbers. You can either use regular numbers or you can use Roman numerals like the 1 or the 1 line and the 2 lines. I don't care. But just keep track of mitotic prophase, metaphase, anaphase, telophase, and meiosis prophase, metaphase, anaphase, telophase 1 and 2. So just keep that in mind when you're sitting. Don't forget those numbers. Okay, so some similarities, right? I said we were going to talk about some similarities and differences. So prophase 1 is very similar to mitotic prophase in that your chromosomes are condensing, the nuclear envelope is breaking down, the nucleolus disappears, your centrioles are spreading apart, your spindle is starting to form. So all of that should be very familiar to you as you will have gone through chapter 9, the mitosis chapter already. The new thing, a new event that's happening in prophase 1 is what we call crossing over. Okay, so you have, I'm going to do some PowerPoint art here real quick. So let's say you have your replicated chromosome, right? So here's, let's say that's chromosome 1 from mom, and we'll do chromosome 1 from dad. And so these are homologues. They're homologous pairs. So they're hanging out next to each other in prophase 1. Okay, so what we actually do is in this process, there's a little region called the chiasma or chiasmata. We actually break off little pieces of each of those chromatids, the sister chromatids, and swap them with our homologous partners. So what that means is I'm going to end up with red chromosome that on the green chromosome, and a little bit of the green. I know I've kind of overlapped it. So if I were to draw this separately out here, I would have something that looks like this. And with my green chromosome, I would have something that looks like this. Okay, so the chromatids in that replicated chromosome swap little pieces of DNA, and it's random. It doesn't always have to happen, and the amount of exchange is different every time the cell goes through meiosis, as you are generating your eggs and sperm. So this is one of the main events that happens in meiosis that allows for that genetic diversity, is these chromatids are going to be swapping some genetic information with their homologous partner. I like to say they're swapping chromatid bits, right? That's funny because it's chromatids and it's a tidbit of the chromosome. All right, so this is a process of what we call recombination or crossing over, and it happens in prophase 1. All right, so in metaphase, remember metaphase, things line up in the middle. We have the same idea, but instead of all of the chromosomes lining up on the middle of the metaphase plate like in mitosis, the partners, those homologues, are going to be lining up on the metaphase plate. So again, let me get my little PowerPoint art going here. So here's my green and red partners here. I'm going to line those up on the metaphase plate. And then let's say I have a short little chromosome down here, and then its partner over here. Okay, I'm going quickly, so I apologize for my bad chromosomes. So then they are going to be connected to their centromeres, and they are going to be connected to these spindle fibers. And we will have our little exchange, right? So we had our crossing over bits here. Let's say we did some crossing over here, and let's say we did some crossing over here. So that's being shown right here. We have the crossing over of those homologous pairs and the crossing over here. So they're calling them recombined chromatids. That's the recombination event. And so in meiosis 1, metaphase 1 of meiosis 1, the homologous pairs line up on the metaphase plate, not like in mitosis. So in mitosis, I'll just do this really quickly, kind of over here, every single chromosome lined up on the metaphase plate. And then every single chromosome was hooked to the spindle fibers, like this, because our goal in metaphase of mitosis was we are wanting to pull apart the chromatids because we have identical copies of the DNA that have to go to all the daughter cells. Well, in meiosis 1, we are trying to reduce the number of chromosomes. And so by splitting the homologues, we are going to accomplish that, right? So let's keep track of our chromosomes here in our cell. So let's see, I'll do a light blue because that's our centromere. So if you count the centromeres, so there's 1, 2, 3, as I'm coloring the centromeres, 4. So we have 4 chromosomes that we started with in this particular cell. And now all the homologues are lined up on the metaphase plate. Let's take a look at what happens next. So anaphase comes after metaphase, same with meiosis. This is anaphase 1. So take a look at what's happening to our homologous chromosomes. They're being pulled apart from each other to opposite ends of the cell, but they're still in that replicated form. So if we're going to count our chromosomes, here is our 1, 2, 3, 4, and we can see they still have their recombined tidbits, if you will, kind of going with them to the opposite ends of the cell. So that's anaphase 1. And telophase 1 is very similar to telophase in mitosis. So the chromosomes start to uncoil, the spindle breaks down, the cell starts to pinch, we get that cleavage furrow. We may or may not get a full nuclear envelope because it's going to jump right into prophase 2 here in a little bit. So that's a slightly variable step is in between telophase 1 and prophase 1. So let's just say telophase 1 and goes to cytokinesis and we have 2 daughter cells, but take a look at how many chromosomes now we're going to have in each of those daughter cells if we're keeping track. So I'll get back to my blue. So here's 1 and 2. So in each of the daughter cells, we now have 2 chromosomes. They're still replicated, but that's just 2 chromosomes. We start off with 4, we now have 2. So by the end of meiosis 1, we have successfully reduced the number of chromosomes by half. Okay, and we have some genetic variability because of that recombination event that happens in prophase 1. Okay, so now we're on to prophase 2. We have 2 completely separate cells. So it's showing we did get our nuclear envelope back a little bit. Our centrioles are going to replicate. So now we're back in prophase. So we're going to see any nuclear envelope that formed is going to break back down. Any unwinding of the chromosomes, they're going to wind back up. The breaking down of your microtubules in telophase is they're just going to start right back up. So we're back in prophase, all of those things that happen in prophase. And again, we'll keep track of our chromosome number. We have one purple one and we have one yellow one. So we have one of each of the kinds. We're now haploid because we had 2 of the big ones and 2 of the little ones. And now we only have one big one and one little one with some genetic variability in there. All right, so this is prophase 2. We're kind of getting ready for these replicated chromosomes to line up on the metaphase plate. So that gets us to metaphase 2 of meiosis 2. So this is going to look a lot more like metaphase of mitosis, where it's the replicated chromosomes that are on the metaphase plate, because now we have to pull apart those chromatids, those replicated pieces. So this is meiosis, metaphase of 2 of meiosis 2. And then anaphase 2, very much like anaphase of mitosis, where now we are pulling apart the replicated chromatids to the opposite ends of the cell. So that's very similar to what we saw in mitosis. And then telophase 2 is like telophase of mitosis. We are going to uncoil the DNA. We are going to wrap the nuclear envelope around the newly separated chromosomes. We are going to break down the spindle, start pinching in with that cleavage furrow. And so we will eventually end up with 4 daughter cells that have half the number of chromosomes and they're genetically unique. So here we can see we have one of the tall and one of the short or medium size, the purple and the yellow. I forget what we started with. I have the whole picture coming up next. So here are your 4 daughter cells. You have, so one parent diploid cell gave you 4 haploid daughter cells, and they are genetically unique because of that crossing over event. So let's take a look at it all kind of together and point out some of those similarities and differences. And we'll have a table that does that also between mitosis and meiosis. So in meiosis 1, prophase 1, the key feature there is that event of crossing over, right? Our chromatid bits are going to be exchanged as those homologues are kind of pairing up in prophase 1. In metaphase 1, we have our homologues lined up on the metaphase plate. Now, I'm going to stop here because we're going to see this a little bit. I didn't mention this when we were doing individual metaphase, but if you'll notice, you have these homologous pairs are lined up different between these. It's only a cell with 4 chromosomes, but we have the purple on this side. Let's get my color. So I have a purple chromosome on this side and then a yellow chromosome on this side. Okay, so that's one arrangement, because as they're going to be pulling apart, that purple is going to lead to 2 of the daughter cells. But the next set of chromosomes, I have yellow on this side and I have purple on this side. Well, that's different, right? How come it can't be purple, purple and yellow, yellow? Well, this is another one of those events that leads to genetic diversity. It's what's called independent assortment or random alignment of the homologous chromosomes. So it could be all different ways, and we're going to see that a little bit later. I just wanted to point out that's where that event occurs. So you have prophase 1 with recombination and metaphase 1 with independent assortment, and that's going to help with this genetic diversity. All right, anaphase 1, we're pulling apart the homologues. Telophase 1, we're splitting into 2 daughter cells at the end of meiosis 1. Meiosis 2, we get our prophase, nuclear envelope breaking down, spindle fibers starting to form, DNA chromosomes condensing. Metaphase 2, all of the replicated chromosomes are lined up on the metaphase plate. Anaphase 2, we're pulling apart the chromatids. Telophase 2, we're splitting all of these cells by cytokinesis, telophase is starting to split, that cytokinesis will finish the split. Nuclear envelope is going around the DNA, it's uncoiling, spindles breaking down, all of the stuff with telophase that we saw before, and then our 4 haploid daughter cells. So we went through it stage by stage, and now you can see it all in one beautiful picture. So as you are working through and studying mitosis and meiosis, make sure you are comfortable knowing the differences and similarities between both of these processes, when they're used, why they're used, where in the body they might take place. All right, so we have this nice kind of handy table that breaks that down a little bit, comparing mitosis and meiosis. So mitosis happens in all of our body cells, right, our autonomic somatic body cells, whereas meiosis only takes place in our gonads, our ovaries and testes, which will be making the sperm and eggs. The final chromosome number. In mitosis, you start with a diploid cell, you will end up with a diploid cell. It is the same. You have 2 copies of every chromosome. You're going to have your homologous pairs. In meiosis, you end up with a haploid daughter cell. You start off diploid and you end up haploid. Mitosis gives you 2 daughter cells. Meiosis gives you 4 daughter cells. We have number of cell divisions per DNA replication. Mitosis, you just get 1 cell division. Meiosis, you get 2. And in the function in animals, since we'll use ourselves as this example, mitosis, if you remember, development, growth, repair, maintenance of tissues, and asexual reproduction, even though animals don't really do that. I would probably not put that in there, maybe protists. And then meiotic cell division, the only function in animals is for sexual reproduction in the making of these sex cells, the gametes. Okay, so here, that was a table showing you some similarities. So in your book, you also have this diagram that kind of goes with it. Let's see where that is on page 149 of the new edition of the textbook. So you can kind of they've lined it up for you. There's no real comparable stages of meiosis 1 to mitosis. So that's why you kind of have that big blank arrow. So all of meiosis 1 is unique to meiosis, in that this is where we have the homologous pairs crossing over, we have the independent assortment, we have the pulling apart of the homologous pairs, and then when we pick back up with meiosis 2, it actually follows very similar to the things that we saw in mitosis, where you are lining up the replicated chromosomes, pulling apart the chromatids, and so that's where you kind of see a little bit more of the similarities between mitosis and meiosis, is with meiosis 2. Okay, so meiosis is essential for sexual reproduction. That's kind of the foundation of this whole idea of being able to create genetically unique offspring. So in meiosis, we get those gametes, the egg and the sperm, that have half the number of chromosomes. Well, why do we have to do that? Can't we just take, your genetically unique mom DNA and mix it with dad DNA? Well, if we did that without meiosis, you're going to have an egg with 46 chromosomes and a sperm with 46 chromosomes. And if I do my math, that's going to give you a zygote with 92 chromosomes. Animals don't. Plants can do that, right? We talked about polyploidy, but animals just don't work that way. And so if we wanted to have success with sexual reproduction, we have to have a way to reduce the number of chromosomes. So when the combination of those unique different individuals combine to give you a unique offspring, we have to reduce that chromosome number by half. So meiosis allows for that. And then because of that, prophase 1 and metaphase 1 events of crossing over and independent assortment, it increases our genetic diversity. So here, just a very basic schematic of your, so maybe this is your egg, parent cell, we'll say egg stem cell. It's actually called an oogonium, which is a nice fun word. And this would be the sperm stem cell. So both of these start off as being diploid. So in your ovaries, if you're female, in your testes, if you're male, you have diploid stem cells that will eventually go through meiosis to produce their haploid daughter cells, which are the egg or the sperm. So then through meiosis, this is the actual egg that has half the number of chromosomes, right? So that's what's ovulated on a monthly basis by females. And then this haploid daughter cell of the sperm stem cell is the sperm. So males start producing sperm at puberty, and they can continuously produce sperm throughout their lives. Females start releasing their oocytes, their eggs, at puberty until menopause. And so when you get sexual intercourse or some combination like in vitro fertilization, if you get an egg and a sperm fusing, it is called fertilization. And those two 1N gametes fuse to make a new diploid organism that's going to have a full complement of both sets. So now that offspring has homologous chromosomes and all of the genetic information needed to grow into that new organism, which is going to be done by mitosis, right? Growth and development. You start from a single fertilized egg and you go to a multi-trillion cell organism thanks to mitosis after you fertilize with the two products of meiosis, right? So there's some connections there. All right, so just to review, when specifically does the genetic diversity of meiosis happen? So I've mentioned a couple times, there's a reason for that. That's an important idea. Hopefully you're kind of catching on that crossing over and recombination in prophase 1 and the random or independent assortment of homologous chromosomes in metaphase 1. Those are the events of meiosis that lead to genetic diversity. Okay, so your egg and sperm are unique because of those things. So here's kind of a cool thing to think about. I usually use males as an example because they produce way more sperm than females do eggs in their lifetime. So a normal ejaculation has about 250 to 500 million sperm per ejaculation. All right, and what's cool about this, because of those two events, the recombination and the independent assortment of meiosis 1, every single one of those sperm is genetically different than every other single sperm that man will produce in their entire lifetime. So think about all of the ejaculation events that a man has from puberty to death, and it's 500 million sperm per ejaculation over a whole lifetime, maybe 80 years, 70, 80 years. Of ejaculations, and every single one of those sperm is unique, like a snowflake, right? And female is going to have the same thing. They just don't have quite that quantity of eggs to be released during their lifetime. Okay, so here's a closer look at those genetic diversity events. So the recombination, here's our homologous chromosomes. Gene 1 are the green, gene 2 are kind of like the bluish gray. So the homologous chromosomes, they're replicated. So they've been replicated in the S phase. We are now in prophase 1. They go close together. They kind of sidle up next to each other. And then there's that crossing over of the same gene. You're not going to swap the blue ones with the green ones. They're not going to like reach their chromatids apart. They're always going to be the neighbors, that the same location of the gene on that chromosome. And so then here is the result of that recombination right here. And then if we were to follow through, so in anaphase 1, the homologues get pulled apart, in anaphase 2, the chromatids get pulled apart. So if we look specifically at each one of these chromosomes, they're all say chromosome number 1, right? So we'll say the purple and the yellow are homologous chromosomes number 1. We have chromosome number 1 with a light green and a blue. We have chromosome number 2 with a dark green and a blue, half maternal, half paternal. Then we have chromosome number 1 that's got light green and a blue, but it's not the same as this one up here. So this one and this one are not the same because this is half maternal and half paternal. And then we have a dark green and a light blue, but again, it's not the same because this one was all purple and this other one, right? So these are the most similar, but they're not because they have different combinations of those alleles, whether the mom or the dad's gonna have a dominant or recessive or both or whatever, that gives you that genetic diversity in the recombination events. The other one that we're focusing on is that random assortment, also known as independent assortment in metaphase 1. So it is how those homologous chromosomes are lined up on the metaphase plate. So in this example, they're just giving you an organism that has 6 chromosomes. It's got 3 pairs, 3 homologous pairs. You could have on this top one, it's showing all purple on the top and all yellow on the bottom. Okay, so that could be one combination, this combination right here. So this combination, if that was to go through meiosis 1 and meiosis 2, down here, these would be your daughter cell combination. So this would be the end of meiosis 1. We've separated our homologues, all the purple in one cell, all the yellow in the other, and then just half of our daughter cells are shown here. All of the daughter cells would be purple, 2 of them would be purple, 2 of them would be yellow. Okay, so that's just one possibility. In this next combination, this one right here, we have 2 purples on the top and 1 yellow, and then 2 yellows on the bottom and 1 purple. So if we go through meiosis 1, we can see how there is some differences. These are not equal. We have 2 purples and 1 yellow, we have 2 yellows and 1 purple, and then if you go down to the end of meiosis 2, we can see how those chromosomes are going to be different. And then you can just keep going on. So here we have another combination, and then we have another combination, and then we have another combination and different combinations. So for comparing all of the daughter cell outcomes, we can see how there's variability. Now that's a lot of combinations, and that was just for an organism that had 3 chromosomes, 6 chromosomes total, 3 pairs of homologues. How many do we have? We have 23 pairs. So there's probably some mathematical combination of what are all the different permutations of your maternal and paternal homologous chromosomes lining up on that metaphase plate. It's just an astoundingly huge number. So those are a little bit more, that's a little bit more detail of our prophase 1 and metaphase 1 events. Now, I didn't put this in the slide, but I wanted to bring it up. Your book mentions it. I think it's got some statistics that I can take a look at. So yes, this random crossing over and this random assortment give you genetic diversity of your egg and sperm. So true. But there's one other layer of genetic variability that can take place. What's the likelihood, not the likelihood, that's the wrong word. So if you are a couple and you're conceiving and the woman ovulates her egg, it's got that particular combination of genes because of the meiotic event. But then the sperm, there's 500 million sperm, not all of them close don't get to the egg, but let's say 100 of them reach the egg. And it's the first sperm that penetrates through the egg wall to fertilize that egg. So out of those 100 sperm, and they're all genetically unique, that combination of that egg and that sperm gives you that organism, right? So if you think I have one son and I'm like, okay, so if it was a different packet of genes, it was a different sperm that fertilized that egg at that particular ovulation event, I could have a completely different child. He could be a boy, but have completely different appearances or personality traits. It could be a girl with completely, maybe similar personality or a different personality. So it adds to that level of genetic diversity is the randomness of which egg and which sperm are going to be used in that fertilization event. So it says, therefore, the chances that your parents could produce another child who is genetically identical to you are about 1 in 64 trillion times. Factoring in some of that genetic, these events that are happening. So it's astronomical. It's, no wonder why we don't have a whole bunch of clones running around. The only natural clones are twins, identical twins for that matter. Okay, so we're gonna wrap up this chapter taking a look at what could happen if there were some errors that occurred during meiosis. And it's true, it does happen. But most of the time, those chromosomal abnormalities lead to natural abortion or miscarriage. So the woman may not even know she was pregnant, or there could have been a miscarriage very early on, because when animal cells, especially humans, have a inappropriate number of chromosomes, that cell is usually not viable. So there's very few examples of where you can actually have different numbers of chromosomes outside of our normal 46, where the offspring is actually viable. So about 20 to 50% of miscarriages are due to these chromosomal abnormalities that are happening during meiosis. It's a process called non-disjunction, and it's where the anaphase event does not occur properly. I'm going to show you a picture of that here next. But there are a few examples where the zygote remains viable and they grow up and they're individual. I hate to use the word normal versus abnormal, but some of them have normal physical characteristics and some of them have abnormal physical and mental characteristics. So we'll take a look at those. So here's what non-disjunction means. So on the far left, this is your normal meiosis. So you get normal separation of homologues in meiosis 1, and you get normal separation of chromatids in meiosis 2. To get each one of your daughter cells has a simple complement, their haploid version, one of each chromosome. Now take a look at the middle column. This is non-disjunction during meiosis 1. So remember, meiosis 1 and anaphase, we're pulling apart the homologues. Now take a look at this right here. Okay, so what's happening is that second set of homologues, the larger of those two chromosome pairs, both of them get pulled to one side. So what that means is this cell over here is missing its chromosome, and this cell over here has too many. So it's got one's got an extra chromosome and one is missing a chromosome. So then if they go through and continue and finish up meiosis 2, take a look at your daughter cells. You have half the cells have one too many chromosomes, and half the cells have one too few chromosomes. Now these are the products of meiosis, or egg and sperm. So these could be an egg, these could be a sperm, and if they get fertilized with another normal egg or sperm, then we're going to have an abnormal number of chromosomes. So let's take a look at the last column. This is non-disjunction of meiosis 2, which was the separation of the chromatids. So our homologues get separated fine, so we're good up to this point. But take a look down here. So it's in this cell, or this part of that daughter cell, where that yellow replicated chromosome doesn't split properly. And so here, in this case, half the daughter cells are normal, so they're good. They're just haploid. It is the daughter cells for this guy right here that have an abnormal number of chromosomes. So let's, for example, I'll take one of these. We'll do that last one. So we've got this. So let's say this is the egg. It has 2 of these chromosomes because it was the yellow ones that didn't separate, and it has 1 purple. Okay, so that's this guy right here. So let's say that is the egg. And then here comes a sperm with its normal complement, right? So we have the 1 purple and 1 yellow. That's what we're supposed to end up with. That's normal. So if this is the sperm, it's got the little flagella, so this is the sperm. If these 2 fuse, we're going to end up, so this would be my zygote, right? So take a look at what those chromosomal combinations are going to look like. This individual will have 3 of the yellows, which is an abnormal, and 2 of the purples, which is normal. So they're going to have an extra of that large yellow chromosome. So when you have extra chromosomes, it's called polyploidy, right? Or if you have 3 of something, we can be a little bit more specific, it's called trisomy. If you are missing one, so if we would have done this one down here, it's -1, then you would have only 1 copy of something, and that's called aneuploidy, or monosomy, means you only have 1 copy of it. So your book has a few examples of these dealing with chromosomal abnormalities of your X and Y chromosomes, because those are the ones that have some ability to be normal for the most part, these viable offspring. So if you have, I'm just going to focus on this column here. So if you get an X chromosome and no other X chromosome, or no Y chromosome, you just have 1 X, you're a monosomy of your X chromosome. This is called Turner syndrome. And the females are fairly normal. They have some physical characteristics. They might be short in stature. Sometimes they're sterile, like they won't be able to reproduce. Sometimes they can. So that is Turner syndrome. If you have extra X's, so let's say there was non-disjunction in one of the sperm and you get 3 X's, this would be a female, and it's called trisomy X. And for the most part, they're relatively, I want to say phenotype, but you guys don't know that word yet. We're going to be learning it in chapter 11. So phenotype is your physical expression of all of your traits. So their phenotype is normal. There's really no necessary indication that they have a different genetic makeup for their sex chromosomes. There could be some non-disjunction and you get what's called XXY, this one right here. XXY is called Klinefelter syndrome. So this is males that they're male in physical or biological sense necessarily. They usually have testes and scrotum and penis. But they might have some more feminine physical characteristics, usually taller, thinner. They might have larger breast tissue, very little facial hair. They might have broader hips, and sometimes they're sterile. So a Klinefelter male is a male biologically because they've got the Y chromosome, but they might have some more feminine characteristics. And then we have the last one where you might have XYY. So these are males. They have the Y there. The Y kind of gives you the maleness. But that extra Y chromosome really doesn't do too much of a difference. It's kind of like the trisomy X. That extra sex chromosome doesn't really affect your outward appearances and your phenotypes. This is called Jacob syndrome, and there might be some men out there that never even know that they're going to have that case. Okay. And then the last chromosomal abnormality we'll talk about is with autosome number 21, not a sex chromosome. This is just chromosome number 21. There's an extra one of those. It's called trisomy. So there's 3 of them. You can see them here in the karyotype. So this is a male, right? So there's 2 X, oh, sorry, not male. So this is a female. So you have an XX chromosome here. Here's chromosome number 22. Here's the 2 X's. So this is a Down syndrome female, and there's maybe hers are representing that picture of that little sister there has Down syndrome. So this is one of the few chromosomal abnormalities where the baby can actually be viable and grow and live without severe deformities that might show up with other chromosomal trisomies or monosomies that would cause a natural miscarriage due to just it's too much of an error in the chromosome number. All right, so that finishes up our chapter on meiosis. So I put a picture of a diverse human race here. We are all of the same species, but we are all different because of those events in meiosis. The crossing over, the independent assortment, and the random fertilization events of which sperm is going to fertilize which egg. So it's kind of a nice way to end on taking a look at the diversity of life itself. So we'll be seeing some of these terms and topics as we continue on into inheritance and genetic characteristics. So I will see you next time.